One. Good afternoon. On behalf of Mates Institute of Pharmacy, I am Dr. Santosh Chajet, Associate Professor. I welcome you all for third session of the day of the FDP, New Age Educational Standard for the Teachers. I am delighted to introduce our next speaker, Dr. Akshay Shrivastava sir. Sir is currently working as an associate professor in the Department of Medical Devices at the National Institute of Pharmaceutical Education and Research, NIPER, Ahemdabad. He is working in the area of biomaterial design and fabrication, tissue engineering, in vitro models, and bioseparations. He has secured DST, SERB Early Career and Core Grant and was honored with the prestigious award of Ramalingaswamy Re-Entry Fellowship. He has worked on two industrial projects and have been involved in the nurturing startup at NIPER. His innovations have been acknowledged by receiving one patent and other three have been applied. Sir has published more than 40 research papers in the Journal of Repute, and he has authored 5 book chapters with total citations of 1600 plus. With this brief introduction, I request Sir to enlighten the audience with your words of wisdom. Over to you, Sir.
Thank you very much, Dr. Chajjar, for the nice introduction. And I also would like to extend my thanks to all the members at MET Nashik for inviting me for this lecture, where I would be like to show cause the new areas in the medical devices and particularly focusing on the spinal cord regeneration. So let me share my slides. Just a moment. Yeah, I hope it is visible.
No, sir. No. Now it is.
Yeah. All
Fine,
right.
fine.
All right, everyone. So in today's session, I am going to talk about the implantable medical devices for spinal cord regeneration, and I have also received a grant from DST, Department of Science and Technology, Ministry of Chemistry, Ministry of MHRD has given us a generous grant to carry out my research work in this particular area. So spinal cord injury is a very serious concern worldwide. Okay, and it has a very high socioeconomic burden that actually significantly affect the patient. Okay, and if you look at the various causes of the spinal cord injury, the most commonly this injury is caused by the vehicular accident, followed by falling from the height, and followed by the sports and other medical surgeries, and it can leads to the paralysis, which is actually a irreversible loss of the locomotion ability. If you look at the Indian scenario, almost 15,000 cases per year is reported. Okay, if you go internationally and worldwide, you will find the ratios are further multiplying into a very high number. And if we try to understand that why it leads to irreversible loss in the locomotion, we must understand the pathophysiology and what are the various challenges why the spinal cord does not regenerate. So spinal cord pathology is multifaceted, and it mainly uh, attenuated because of the inflammation. And the another thing which we all probably know, that the axonal regeneration capability of all the neurons are very limited. Hence, if we can somehow enhance the regeneration capability of the neurons, at the same time, if we can suppress the inflammation at the site of action, then probably we will be able to regenerate this entire spinal cord in no time. If you look at the injured area of the spinal cord, then it leads to many pathological changes. For example, most prominently, it causes the astrocytes to become reactive. The astrocytes are the cells which lives in the spinal cord. They become reactive upon injury. There is a macroglia and macrophages like cells which again become reactive, and they start showing inflammation. So these two cell types are majorly involved in creating an inflammatory microenvironment after the injury in the spinal cord. Meanwhile, there are other chemical processes which causes the formation of reactive oxygen species, which is again detrimental for the tissue. Overall, these tissue try to regenerate over a period of time. Okay, on its own, causes fibrosis and leads to scar formation. Okay, and thus it has a poor intrinsic regeneration ability. So there are various treatment approaches which are available, and these therapies include biomaterials that fill the cavity and injured area at the site, deliver the drugs, that's a pharmacological approach, transplant the cells or stem cells either alone or with the scaffold, that may help in trying to repair and regenerate. Apart from that, there are either alone or in combination with other therapies, the surgeons and the doctors give the spinal stimulation. So in this particular stimulation, they give current also. So we know that neuronal cells are very good conductor of electricity, and there is a very minute amount of current, or you can say potential, generated in the spinal cord and neuronal cells. So it is seen that spinal stimulation also enhance the regeneration, or you can say the repair process. However, they all are currently associated with certain limitations, and still the repair and regeneration of a spinal cord is tricky. Okay, in only rare situation it is achievable. For example, the rate of success for spinal cord stimulation, it's only up to 50 to 60%, and per patient has to sit on certain chair every day. It has to go to get the stimulation, so patient compliance is less. And if we talk about the pharmacological, it is actually uh, mainly relieve the pain, and even if there are some drugs available, they try to regenerate the axons. However, multiple dose is required, and it does not show a good axonal regeneration property. Then we comes to the cell therapy. So cell therapy is like many groups and the clinicians are trying to explore the stem cell therapy, and but they need to identify the unique stem cells. They need to find the unique stem cell. They are trying to use one of the source for bone marrow. So bone marrow stem cells are used, but again the success is limited, and it is a time consuming method to isolate the cells, to maintain the cells, and then deliver the cells. Okay, and of course these stems are isolated from the another patients, or another, not patient, another healthy individual, and there are some ethical constraints also there related to these stem cells usage. When I was starting this project, so I also visited one of the local hospital at Ahemdabad, and the professor name is Professor Aruna Varinkar, and we I has interacted with her in one of the presentations where she has showed that BMSC's delivery is actually trying is actually helping in regeneration process of the spinal cord. However, after the interaction we got to know that there are still a limitation, because the uh, at the injury site the inflammation is very high, and because of these inflammation, stem cells are either differentiate into different lineage, or they get killed and undergo apoptosis and got death. So the other way is, we give stem cells in a particular scaffold that probably could protect stem cells. Because there is another limitation which is associated with this cell therapy is the cell leakage. You implant the cell at the site of action, but the cells, they are in so many high numbers, it does not retain at the position and they leak out. So hence its regeneration capability is limited. Also, you require a very high number of, like in millions, you need millions of cells. And stem cells having a progenitor, like having a regenerative capability, they are present in very minute amount in the adult tissues. So the extraction of or isolation of large number of stem cells is also one of the limitation. Hence, if then the new field came up in the form of biomaterials, and these biomaterials are of different forms, and in particular, if I talk about the spinal cord regeneration, mainly two types of biomaterial scaffolds have been utilized widely. One is the hydrogel type and the second is nanofibrous type. Both these material claim to mimic the extracellular matrix which is present in spinal cord. Okay, and these hydrogels and nanofibers claims that it can uh, fully regenerate if mixed with the stem cells and given as a treatment. However, these materials also suffer from some of the limitation till date, and there is a like dead end of the pores. So the pores are blocked. They does not allow the regeneration of the entire tissue. Okay. However, there are many advanced technique now which can overcome this problem. So this is not a very big problem at the moment. There are many processing techniques are available, but they also lack the 3D cellular organizations. If you mix it with the stem cells with the porous material, biomaterial, then proper 3D cellular organization cannot be achieved. Also, there are reports that they do not suppress the inflammation and it takes a very long time for the regeneration. And till date, the biomaterial based treatment has shown that average time of axonal regeneration is going much above than the 8 week. Okay, and only partial recovery is possible. So, with this gap in the design of biomaterial and the regenerative capability of the biomaterial, many scaffolds have been developed. You see, spinal cord is a cylindrical tissue, so you can have something like a hollow cylinder made up of biomaterial where central portion is remain hollow, which allows the growth of the cells, and in the surrounding wall, either stem cells or some growth factors can be incorporated in this material, which helps in the regeneration. uh, In some of these studies, a multi-channel conduits are also developed as an implant, which also which has also shown to help in the repair and regeneration. There is a company called In Vivo Therapeutics, and they have developed a neurospinal scaffold, which looks like a porous scaffold, and they are mechanically compliant, three-dimensional, porous, and biodegradable scaffolds which they have developed, and it has shown some success in recently. However, they are still not able to achieve the complete repair based on the implant and the implant plus cells. There are groups which are working on developing aligned nanofibers as a fibrous material. It has shown a very good potential in terms of cell alignment and the cell adhesion. However, these materials also show a limited success in terms of recovering the locomotory ability of these of the animal models where they have tested. So most of these materials are based on collagen. It is most desirable polymer for spinal cord regeneration. There are other polymers also used, PLA, PLGA, and other combinations of synthetic polymers have also been used. In some cases, alginates are also reported. However, out of all these, it has seen the collagen type of material is actually provide a appropriate mechanical and biological cue which helps in the repair and the regeneration of the spinal cord. So, with this idea, what we got to know so far that you know uh, there are several properties which one needs to adopt before applying for the spinal cord regeneration, and we can how we can accelerate the axonal regeneration. So the material should have porosity. It should be three-dimensional in nature, in shape, and provide a porosity, a porous network through which the cells can try to migrate and connect with each other in a broken spinal cord. The material should have a sufficient mechanical elasticity. It is seen that there are several stimulus which are important for the spinal cord. Okay, one of them is the mechanical sponginess or mechanical stimulus. Similarly, electric conductivity. So you see the spinal stimulation therapy. If you go to the physiotherapy, most of these spinal cord injured patient, there is a prolonged physiotherapy is proposed by the surgeons and the clinicians. It is because they want to stimulate the microenvironment at the site of injury with either mechanical pressure or by electrical pressure. Okay, or in many cases, both kind of pressures are used as a physiotherapy protocol. Okay, so we also understood that probably these two are very important properties a spinal cord regeneration conduit must have. It also, from literature and in our personal experience through the various experiments, we found that suppressing the neuroinflammation, and particularly I talked about the microglia and the astrocytes. So these two cells should also be modulated appropriately, which supports the stem cell, which supports the spinal cord regeneration. Now, if you look at the entire components of the conduit which is required, it it has a stimulus, it has a porous network, but at the same time, it should also support the stem cell proliferation, okay, and support as a matrix for the stem cells delivery, okay. So hence we propose a solution as electroconductive, mechanically spongy, and super macroporous material, which may suppress the neuroinflammation and promote faster axonal regeneration in spinal cord injury. So our aim is to have all these combination of properties in a material which can actually promote or accelerate the regeneration of axons. Okay. So for that, we have chosen collagen type 1. I have told you it's a very promising and very demanding material for spinal cord regeneration. And it has a certain stimulus, both as a mechanical as well as electrical. However, the electrical and mechanical properties are not exactly matching up to the mark of the spinal cord. Thus, we have added the graphene material in the starting. And we thought by adding the graphene, probably it will be able to make a robust material which will have a very good, excellent electric conductive property, because graphene is known to have electric conductivity properties which will imparted in the scaffold. It also actually modulates the mechanical properties of the material. Okay, but then we have again gone to the literature and we found that these graphene materials have also shown to enhance angiogenesis. Okay, and it avoid the scar retractions. These graphene and graphene based materials have also shown that it limit the astrocytes reactivity, it controls the macrophages infiltration and inflammation when it is implanted in rat hemisection model. So we have got another important property of graphene, which probably we can exploit in our material, is the modulating of the inflammation. Okay, so with this all information and the combination of property which we are using, so we mixed the graphene with the collagen, but we have not simply made a composite material. We have actually made a cross-linked material. Okay, for the cross-linking of the material, we have generated amino functionalized groups on the graphene, and then these amino functionalized groups were cross-linked, used as a cross-linker to cross-link collagen. And the entire reaction was done in the presence of carbodimide and anhydroxysuccinimide at sub-zero temperature. Okay, and this making this and cross-linking this at sub-zero temperature is actually a protocol which is adopted from the cryogelation technique. So the cryogels are nothing but a hydrogel. It's just simply conventional hydrogel which is synthesized at sub-zero temperature. When you synthesize the cryogel or hydrogel at sub-zero temperature, and mostly these hydrogels are synthesized in water as a solvent. So at sub-zero temperature, these water freezes. When these water freezes, then the local concentration of this polymer becomes so high. It becomes so high that in the small regions, they never freeze, and that is a colligative property. That which means that as you increase the concentration of the solute, there is a depression in the freezing point. So most of the water is frozen, but at some localized part where this polymer and graphene becomes in very high concentration, it does not freeze, and there the cross-linking happens. And that's how it forms the boundaries of the gel. Okay, you can see this after 12-15 hours, we can take out the entire system outside at room temperature and thaw it in the water. You will see all the ice melts. When ice melts, it leaves behind the pores. Ice has a tendency to connect and to grow, and that's why most of these pores has connectivity and have large diameter. So this is, you see, the cylinder-like material, like a spinal cord kind of a material was developed. In this video, probably I think this video is not working. I'm sorry for that. But what we want to show here is with this forceps, we can compress this gel, this small piece of gel, more than 90%. Okay. And if we release the pressure from the forceps, it again regains its original shape if you dip it in water. So we have used different concentration of graphene to optimize the whole protocol and then further characterize the material. In the characterization, we first wanted to know how much cross-linking has occurred. For that, we have done to check the amine functionalities. We estimated the cross-linking, and we found that 0.5% weight by volume of graphene when mixed with the collagen provides a highest degree of cross-linking compared to the 0.1% and 1%. One can expect 1% should show higher, but 1% graphene start precipitating during the synthesis, and it did not available for the cross-linking, and hence we haven't seen a good material property in terms of cross-linking as well. We have further checked the cross-linking of our material with graphene and collagen by thermogravimetric analysis and dynamic scattering calorimetric analysis. You can see the 0.5% shows much more stability compared to the other concentrations of graphene, which further shows that this material is have a high cross-linking, and that's why probably they are more stable to the temperature. Similar trend we have seen in dynamic scattering calorimetric assays analysis, and again the material is showing the incorporation of both the groups, and 0.51 looks fairly good compared to the only collagen material. So in all our comparisons, we have keeping this EDC-NHS group. This EDC-NHS group is only collagen, no graphene. So in my all further slides, wherever you see the EDC-NHS, it means it's only collagen plus EDC-NHS, no graphene. Okay, that is our like a control, which is collagen hydrogels. We further check the very important property called electric conductivity by using 4-point probe method. and we found that 0.5% graphene compared to other, showing the better current conductivity for current conductivity in our materials. Okay, and the it is significantly higher compared to both 0.1% graphene incorporated and 1% graphene incorporated uh, uh, hydrogens. So with this, like, material has a good and sufficient electric conductivity, and we move further to know what is the porosity. Because I told you that electric conductivity, porosity, and mechanical strength are the three material properties which are important. When we looked at a porosity analysis, so there are several methods of doing that. Swelling in water, then estimating the pore volume and estimating the interconnected volume, and you can observe these pore size in the scanning electron microscope. Overall, we did not found the much difference between all the concentrations in comparing to EDC, NHS, cross-linked collagen. So all of them were equally swellable. Okay, there is not much difference in the porosity. However, definitely there is certain decrease in the pore volume when the graphene is incorporated compared to EDC, NHS, cross-linked, which is definitely possible because we have added another component as a graphene, which is absent. So graphene has taken some area in the hydrogel, and thus the slightly decrease in the pore volume is observed, which is fair enough. They all are having a good interconnected porosity, means all the pores are highly interconnected, and even the same image shows that the porosity is not compromised by addition of graphene. Rather, we will see more distributed and a more distributed and a thick walled porosity is present in case of graphene incorporated. So we further wanted to know, I mean, at what level the graphene has actually influenced the porosity of the material. For that, we did the microcity analysis. Okay, so this microcity analysis is actually given us inside depth about the material properties, and you see most of the material looks same in this binarized image, but when we reconstruct into three-dimensional, you will see alone collagen cross-linked with EC showing more fibrous like nature in the hydrogel. However, these fibrous nature are lost when we have incorporated 0.1% graphene and 0.5% graphene. Rather, these graphenes probably are present as these small islands. Okay, and these are multi-layered graphene which is incorporated, and thus they are having some aggregates. These aggregates are present which may act as a small islands of conductivity. Moreover, the porosity is much more controlled in the case of 0.5% graphene incorporation. The distribution is highly controlled, and most of the pore sizes are of same size. Even you will see the pore wall thickness is slightly higher. It means this thickness, once the thickness of the pore wall increases, it means it imparts to its mechanical property. Okay, we have done some more analysis of microcity, and we found some important figures. For example, the closed porosity is much less in 0.5% graphene compared to the EDCNHS. And likewise, the open porosity is much more in graphene incorporated cryogels compared to only EDCNHS collagen cryogels. Connectivity is also high. The degree of anisotropy is quite low. It's more stable structure. So overall, in terms of porosity and interconnectivity, the graphene incorporated cryogels have shown better porous morphology compared to EDC and NHS cross-linked collagen. So, as you see, the pore walls are little thicker in the porosity analysis, and it was also observed in scanning electron microscope. Then, when the pore walls are thick, then it will affect on its mechanical property, and we wanted to observe. So, we did the rheological studies. And in the rheological studies, you can see the storage modulus of 0.5% graphene is significantly higher, which shows the material is, first of all, very spongy, as well as has a good gel-like material, having a sufficient strength. We have also tested the material in universal tensile testing machine, where we did the compression study, and you see the stress versus strain graph, which is a very, it's like follows the Hooke's law, if you remember. Then this strain versus strain, you see very small stress, it compresses very high, more than 60% of the material is compressed. Okay, at very very minimum stress, you put a pressure a little bit, it will compress. But you see, out of all these, if you further increase the stress, only one particular material is stable, and that is 0.5% graphene. All EDCNHS and zero point 0.1 are broken here beyond, so they cannot bear stress more than 0.2, 0.1. But the 0.5% graphene can bear stress up to 1.2 megapascal. Okay, so, and overall Young modulus, which actually represent the sponginess of the material, compressiveness of the material. So you can see 0.5% is highly compressive and very spongy like material compared to EDCNHS and graphene. If you look at the toughness, how tough is the material? Because of this large area which is covered, and the 0.5% has significantly much higher toughness also. So material is spongy as well as tough. It won't break. It is spongy, you can compress it, but it won't break. Okay, so toughness is also comparatively higher compared to EDCNHS alone. So material has excellent mechanical strength, which is required for the spinal cord like area. So spinal cord, if you talk about the spinal cord, its mechanical strength is somewhere between 200 kilopascal to 400 kilopascal, and our material is somewhere in the middle. Okay, so that is excellent. We are trying to match up the same mechanical strength of the spinal cord. Now, we further want to check these properties, which are chain mechanical properties, affecting, very largely porosity is controlled, the porous shape is different. Then what is happening? How this cross-linking is happening, whether the cross-linking is happening, and how it is affecting the collagen structure, whether the collagen is compromised or not compromised. For that, we checked the graphene spectra in Raman spectroscopic analysis, and we found there is not much difference in the graphene. All the graphenes which are incorporated in 0.5%, 0.1% are same, so there is no deviations between the all groups. However, when we looked at the collagen in Raman spectroscopy, we found there is an uptrend in the regions of amide 3 and amide 1. This amide 3 and amide 1 kind of region are helical regions, which represent the helical region of the collagen. And you see more helical content is present in 0.5% graphene, which is shown by a red graph here. Okay. We further zoomed in this area and looked into it, and we found the amide 3 region is significantly higher, which shows that more helical content of collagen is retained or promoted in 0.5% graphene incorporated collagen hydrogels. So with this, it is also a very informative data, which tells us that as the helical content increases, the strength of the collagen is also increases, and that's why probably we get a better toughness in the mechanical strength. So graphene is also promoting the helical content of the material as per our cross-linking strategy. So we looked into the ratios of various intensity, which is a usual way of looking at it, which talked about the orderliness of the collagen and how much collagen structure is ordered. You see more ordered collagen is present when 0.5% graphene is incorporated. Even though by looking at this Raman spectroscopy, we can further look into the cross-linking also, and for that you need to do ratio of 1660 to 1690 intensity, the wave numbers, and then you will see more cross-linking is observed in the 0.5% graphene. Okay, so cross-linking and promotion of alpha helical content is actually affecting the mechanical properties of the hydrogel. So with this, our material is ready to investigated in vitro and in vivo analysis. So our material has sufficient porosity, have good electric and mechanical strength, and properties. So we further characterize in vitro, and we wanted to see what is happening with BMSCs when we load onto them. Okay, so we loaded the BMSCs. We have given electric stimulus to the scaffold. We wanted to see what kind of growth factors and other paracrine secretions are happening. We also wanted to see when we grow macrophages with this material, whether we can control the macrophages reactivity, because we have to control, as in spinal cord injury, macrophages are very reactive. If we can control that reactivity, we probably will be able to enhance or accelerate the spinal cord regeneration. So we have designed this experiment, and you see this is a device what we have developed in our plate, 6-well plate. These electrodes are inserted in the well between which the material was placed and electric current was given. Okay, after giving electric current, we wanted to see what happened to the stem cells, whether the stemness was retained or not. And we have seen by looking at two markers, CD90 and CD73, we found that the electric stimulus has actually promoted the stemness in 0.5% graphene. Comparatively, it has shown a large deviations, much promoted. So stemness is maintained or increased when the electric current was given in 0.5% graphene incorporated collagen cryogel. We further wanted to check another very important biomolecule called ATP. In some of the recent report, we found that ATP is actually energy molecule. So when ATP is given, it actually helps the neurons and other cells to multiply and repair and regenerate. So we want to see what happened to ATP, whether because our material is conductive in nature, can we modulate the ATP levels just by giving current? Again, surprisingly, which is great data showing that by giving the electric stimulus, we can actually increase the ATP measurement on day 3. It is progressive from day 1 to day 2 and day 2 to day 3. You can see there's an increase uptrend in the increase of the ATP. Hence, we can use our scaffold giving by and then give current, which can probably increase the ATP production in the PMSCs and other cells, which probably will be useful for the repair and regenerative processes. We further wanted to know, okay, the BMSCs are happily living. So I haven't shown the toxicity data, which is so these cells and these materials are not toxic to the BMSCs. It has shown to control the stemness, or rather enhance the stemness. Then we wanted to know whether we can differentiate it into the neuronal lineage or not. For that, we have grown the stem BMSCs in the graphene incorporated cryogels, and up to 7 days we grown the stem cells, and then after that we started giving the electric current. Okay, and you can see that 3 basic markers of neuronal differentiation: MAP2 kinase, beta tubulin 3, and nestin. They all increased. But basically, MAP2 kinase and beta tubulin increase, which is a marker of mature neurons, and you can see 0.5% graphene with electric current showing enhanced neuronal differentiation. So material, if subjected under electric current along with the cells, it shows a better neuronal differentiation compared to only cells. So now our next target was to check what happens to the neuroinflammation. Can our material, because of graphene has its property, we want to check whether our developed material can control the immunomodulation. Okay. For that, first we wanted to create an inflammatory environment. We wanted to create inflamed macrophages, because in the spinal cord injury, most of the macrophages are inflamed in nature. So we inflamed the cell line, raw 264.7 cell, which is a macrophage cell line, and we stimulated with the lipopolysaccharide 10 microgram per ml, and we leave it for 48 hours. After that, we checked whether the cells were inflamed or not. So you see the comparison between these two dot plots, which is for the inflammatory dot plot. So normal raw cells have some these are unstimulated with LPS. So with no LPS, this is a normal expression of the inflammatory markers. But after LPS treatment, you see increase in the inflammatory markers. Several markers are increased. Okay, and what we did next, that we took this raw inflamed cells, then we took the media in which these inflamed cells were growing and taken that media, because expecting that media will have a lot of inflammatory markers. We are trying to mimic the in vivo condition in vitro. And then these inflammatory cytokines which are present in the media, we have exposed it to the BMSC's loaded graphene incorporated collagen cryogel and check the canocurin level. So canocurin level, which actually help in measuring the anti-inflammatory properties of stem cells. We want to see whether stem cells growing in these materials can have these inflammatory properties. So you will see that there is not much significant difference you can usually observe in the in the raw cells which are cultured, and you see when we compare with the normal MSC's media. So overall, the kynocurin is actually showing a significant difference between the 0.1 or 0.1% graphene compared to 2D. So this increased kynocurin level is actually showing that stem cells shows this protective nature against the inflammation. However, stem cells alone or with the materials has no separate effect. Comparatively 2D, it is showing the better effect. Okay, but between the groups there is no significant effect. So it has retained the MSC's property. We further wanted to check another inflammatory cells which are macrophages, and we see that cells are nicely attached onto material in this FE7 images. Then we looked into the polarization of the macrophages, because macrophages also become reactive in spinal cord injury. So there are several markers for M1 polarization and M2 polarization. We have chosen CD86 as M1, CD86 at M1, and this is I think CD163 and CD206 for M2. Okay. So you see CD86 is increased in 0.5% on day 3, but surprisingly, if you grow the culture up to 7 days, CD86 decreases significantly, which means that M1, which is a inflammatory macrophage, pro-inflammatory, this pro-inflammatory macrophages decreased after 7 days when you grown onto the material. However, M2 polarization, which is a macrophage differentiate into the anti-inflammatory properties, then you see at day 7 the material is actually showing upregulation in M2 type of polarization. Hence, it means the ratio between M1 to M2 is shifted more towards M2, which means the macrophages are becoming more anti-inflammatory. Means it is going to help the microenvironment in suppressing the inflammation, and our material can do that alone. We further tested it in immunohistochemistry, and we found similar results both in CD86. You see very diminished CD86, means the M1 polarization is very less. Okay. So in vitro, we have tested our material and demonstrated that material is supporting the growth of BMSCs. It is non-toxic and can suppress the inflammation. We further tested whether we in ex vivo culture, okay, to check the immunomodulatory as well as neuronal regeneration potential of the material. For that, we have taken a slice of a rat spinal cord. It's just a 1-2 millimeter piece, I would say. Okay, and that piece, cylindrical piece of spinal cord, we kept it over the gel. Okay, and allow it to attach and attract and embed along with the material on the surface. And then we wanted to see whether the cells grow inside the material or not, what happens to the microglia population, what is astrocytes happening. You see, this is a real image. Probably you are following my cursor, and you see the slice of spinal cord is kept over the material. Okay, we have used 0.5% because it came out like the best combination of graphene with the collagen. And from now onwards, in my presentation, you will see it is called as GR call. Okay, and EDC NHS crosslink will be called as call. So we have tested various biochemical. We have used various biochemical method to test our hypothesis, and which we started calling organ on a gel. You see, after 7 days, this is nicely attached to the matrix. In this video, which is not working, I'm sorry, but in this video, what we are trying to show that by using a forceps, if you try to remove this slice of spinal cord from the material, it is very difficult. Okay, you can only detach by destroying the tissue. Okay, then we further were interested at how much attachment it has done and why it is so attached onto the material. For that, we did the strain electron microscopy, and we found there's a formation of some growth cones with the tissue and the material. Which means the cells which are present in the tissue, they are trying to attach to the material. Okay, and these growth cone formation was observed. Hence, we first looked into the reactivity of the astrocytes, and you see in this RT-PCR data, you will find that GFAP, which is a marker for reactive exercise. You see, GFAP expression is very high in collagen alone, but graphene incorporated, you see at day 7, it is equal to cell culture, and if you go to day 17, it further suppresses the reactivity. Means it is controlling the reactive nature of the astrocytes, because reactive astrocytes are not good. It leads to the scar formation. Okay. Then we further tested at Western blot level, and we found that the GR call, if you look, the expression of GFAP at protein level is also very, very less compared to the control and the collagen. Okay, hence the material can control the astrocyte reactivity as well. We have looked to another marker which is responsible for astrocyte reactivity, that is Vimenting, and we found almost similar trend, highly significantly less reactivity of astrocytes or Vimenting expression in GR call. We have further tested it immunohistochemistry by looking at the expression of GFAP and Vimentin, and we found that the GR call at day 14 is actually suppressing the inflammation. We also tested the microglia. For that, we are using the same marker, CD206 and CD86. You see, in CD206, a high expression of both GR call and call is present. Okay, and in CD86, you see very less expression, significantly less expression of CD86, which is CD8. If CD86 is low, means its inflammatory properties are less. Okay, so GR call is showing very low, pro-inflammatory properties. And similar trend were observed when we did the immunohistochemistry analysis as well in our ex vivo culture. So the material which were tested in vitro with the slice culture, organ on the culture, we've seen that it can control the microglia, it can promote the M1 M2 ratio more towards M2, which is anti-inflammatory. We have seen that astrocytes are controlled, reactive astrocytes expression is less. We further wanted to know again whether neuronal regeneration capability is there or not. For that, we have grown the slice in the material and given the electric stimulator by using these two carbon electrode. For this is like with the 50 millivolts per millimeter for 5 minutes per day from day 17. So up to day 7, we have allowed this slice culture to attach onto the material, and from day 7 to 14 we have given this treatment of electric current every day, 5 minutes. Okay. What we see, beta tubulin, which is a mature neuronal marker, and we see very high expression of beta tubulin at day 14, significantly high, which further confirms that these materials, along with the electric current, okay, can show a very high neuronal regeneration. And same was apparent in our Western blot analysis as well. However, it is also seen that at PCR level, it shows a lot of mRNA is forming. However, in Western blot level, this GR call at day 14, if you look at the beta tubulin, is very high. However, the new N expression is not so high. Okay, it is almost similar to the unstimulated. But overall, the conclusion is that the material along with the current promote neuronal regeneration much better than the collagen hydrogel alone. We further tested it immunohistochemistry analysis by looking at the beta tubulin 3. Again, high expression is seen, which is mean more neuronization in the electric stimulated and unstimulated GR call, and see the new one is also fairly expressed in the material, which further support the neuronal regeneration capability of our material. Then we want to know why this is happening. So there were very like issues, like just how material alone is actually promoting and doing so many good things. So we know that there are something called as mechanosensitive genes. Okay, uh, probably some of you or most of you probably know that, like there is a Nobel Prize last year is given to the piezo channels. Okay, those are mechanical sensitive channels, piezoelectric channels. Okay, similar type of the channels are there are some mechanical sensitive gene analysis what we have conducted. We thought probably the material property is actually influencing the cell behavior. For that, we have chosen three markers: paxillin, focal adhesion, and vinculin. These are three mechanosensitive genes. And we surprisingly find that paxillin and focal adhesion kinase expression is so high at day 14 in GR core. We could not find much difference in vinculin expression. However, collagen alone showing the higher one in vinculin. But our material is working through the paxillin and focal adhesion kinase expression probably. So with this further strengthen that our material is probably working and showing effect, probably because of the changes what they have done in the mechanosensitive genes. We further tested our material in vivo in a rat in a rat model, and for that these are our group of animals what we have taken. We have taken sham, the healthy one, injury. We having as a control we have given collagen alone, and our tested material which is collagen GR core. So we have done the hemisection. We have done the hemisection model, and in at T10 T11 thoracic region, and we have created 2 mm gap of hemisection where we have implanted the material. Okay, at call and GR call. Then we finally extracted the spinal cord after 28. You see the materials are nicely, embedded, or you can say, joined with the injured spinal cord. You see the injury area still intact in the injury area. So we first tested what happens to the animal. Particularly, we were interested in having their locomotory behavior. So we were very surprised to see that GR call material is actually recovering the entire locomotory function. Okay, at 28 day, though the locomotory function was retained, or you can say was already retained at day 14 itself. Okay, so you see in the injury area, you see the back leg, right back leg, it is paralyzed. It's still not working. An animal is just walking with 3 legs. Okay, with call implant, you see there is some recovery, but still it's limping. The rat is limping. It is not walking here and there just like that. But when we look into the GR call implanted material, implanted rat, you see it is walking nicely with a good gait cycle, and all both the back legs are working fine, and it has retained its entire locomotory activity at just day 28. So we did further, test, and we want to look at the BBB scoring, which is this BBB scoring is to know the locomotory function, and you see right from day 14 you see the uptrend in GR call implanted. So the recovery started within 2 weeks, and up to 28 days it has significantly recovered compared to the collagen and sham or injured area. Sorry, injured alone. This is sham. With just for 28 days, it has shown significant difference. Then we compared that about the recovery rate, and we found a similar recovery with same BBB score. How many different researchers have done so far? We found that there are few material which are reported, and most of them show either 8 weeks, 6 weeks, or 12 weeks of recovery. We can achieve similar recovery in just 4 weeks. Okay, we further tested the material by other grip strength analysis and hot cold plate analysis. You see not much difference in grip strength. It's all same. So these kind of tests are sometimes notorious. You don't get like a clear data. We did hot cold plate analysis, like a withdrawal of power upon temperature, and you see they have retained, they have recovered their sensory motor activities. Okay, so you see the GR call is almost achieved the similar uh, uh, properties or activities just like sham. Injury and call area did not show much difference. So this has further strengthened our material, properties of our material, and can control or increase the locomotory functions upon implantation. For again to demonstrate what is happening, we looked into the sections of the our material, and you see here this is a injury area which is marked by yellow line, and this is like a collagen implanted material. We looked at beta tubulin. There is some expression of beta tubulin in both, as well as neurofilament. But when you look into the GR call, you see very high expression, and it has shown to form the connection between the broken spinal cord. Even neurofilament 200 is so high expression, which is again for the mature neurons. Okay, so this altogether shows that the neuronal regeneration is enhanced by the implantation of GR call in rat model. Again, just to prove our hypothesis in vitro about the inflammation, and you see that the CD206 expression is very high, in GR call, and the call implants as well. And CD86 is also suppressed in GR call kind of material, which further supports that microglia polarization is controlled upon in GR call. We further looked into the astrocytes reactivity, and you see GFAP is again the astrocyte reactivity marker, and you see the GR call is showing very less astrocyte reactivity compared to the injury and the collagen. We also looked into the Vimenting expression, and Vimenting expression is also comparable and demonstrating that GR call actually decreases the both the expression of GFAP and Vimentin in vivo. If it can decrease, it means it is actually suppressing the neuroinflammation. So why this is all happening? So we need some mechanistic answers, and for that again we looked into the literature, and we wanted to know what is all about the stat expression. So we found that there is a stat pathway which is highly modulated, and this stat is actually responsible for modulating the astrocyte reactivity. So you see that GR call showing much less expression of STAT3. More the expression of STAT3, more you will see the inflammation. And you see that probably GR call is decreasing the expression of STAT3, and hence it is controlling the neuroinflammation in this spinal cord injury. So this further confirms that this STAT3 could be one of the pathway. However, we need to do the detailed mechanistic, and for that there is an ongoing study by using next generation sequencing, which and analysis is ongoing. And there, just a preliminary data I just want to share, that it is showing a very clear cut that GR call is actually modulating the inflammatory as well as microglia kind of pathways. And hence, because of this, I guess it is supporting the neuronal regeneration. We further looked into the LBL staining and HNE staining, where the injury area it is marked in HNE staining, and you can see the sham is healthy. And when GR call is implanted, you see the entire material, the cells are infiltrated and try to connect each other. The LFB staining was done just to see myelination, at what extent the myelination has happened, because this is another important biochemical feature of the spinal cord. If you can achieve the same myelination or enhance the myelination of the growing in the growing neuron, you will actually help in the better connectivity. Again, you see that GR call has shown a very high, myelination compared to the injury area. We also wanted to see what the ECM matrix is happening, whether ECM matrix is remodeling, because it's in the spinal cord. The ECM also plays a very important role in maintaining the connectivity between the cells, and also is important to give appropriate stimulus. We wanted to see why our material is alone is promoting. So we have found that cells are modulating. We have found that some pathways are modulating. Similarly, we have seen even the ECM matrix also remodeling, and for that we have chosen two markers which are very prominent in the spinal cord, that is fibronectin and laminin. So both fibronectin and laminin has shown a very high expression. In the case of GR call implanting, you see a very nice fine connections are formed, and the matrix is deposited throughout both fibronectin and laminin. And probably this is one another reason that the surrounding cells are actually started secreting laminin, which is deposited and hence allowed the cell attachment for the complete attachment of the broken spinal cord. We further try to analyze that how cells sense the orientation, how they move into the porous network and connect. For that, we did the cell orientation analysis by using one of the ImageJ plugin, and it was done in in one of the group in Switzerland have done that. So we have used the same protocol, and we found the orientation of cell is also much controlled in GR call compared to control. So with this, we would like to tell you the overall summary of our work, that upon a spinal cord injury, if GR call is implanted, what we observe that graphene probably is actually controlling the astrocyte reactivity and microglia polarization, graphene and collagen cryogens. And because of the interconnected porosity and suppressing the inflammation together, can help in accelerated regeneration. And these were the three major barriers. Clinically, if you know about my spinal cord, the three barriers are reactive astrocytes, microglia reactivity, and the hindered or limited regeneration capacity of the neuron. We are trying to answer all these three with our material, where we can control the astrocyte reactivity, can control the microglia polarization, and can accelerate the regeneration. So this is the end of my talk, and I would like to acknowledge mainly my PhD student, Dr. Gopal, and some other group members as well. There are so many masters who are also involved from time to time in this project. I would like to thank my collaborator, Dr. Hemant Kumar. He was a very, we worked in very close association to particularly in the in vivo work, and I'm thankful to all my funding agencies, including NIPER. I've got DST, both DST Serve and Core Grant I'm also a DBT Ramalingam Swami Fellow, so thanks to that grant as well. And overall, for all the analysis, microscopy, and all those things, I would like to thank the Central Instrumentation Facility at NIPER Ahemdabad. I would like to entertain any questions if you may have. You can contact me if you have any questions later on as well. Thank you very much.
Thank you very much, sir, for your talk. Sir, you have enlightened participants with various aspects of implantable medical devices for spinal cord regeneration. I appeal participants to raise any query if you have. There is a question from Mr. Eknath Ahire, sir. What are the regulatory considerations for the medical devices?
Yeah, I mean, of course, this, if you talk about particularly spinal cord, is a implantable material. It goes inside the body, so they are very stringent regulatory pathway. And I just briefly tell you that medical devices can be classified into four regulatory paths: Class A type material, Class B type of device, Class C, Class D. Class C and Class D are having a medical devices which are implanted inside the body. Okay, Class A is not implanted inside the body; it remain outside, and the regulatory path is quite easier. Class B is also implanted, but depends on what is the extent of its interaction with the body. But Class C and Class D is like require a very robust clinical data. Okay, all the implants, and the spinal cord implant what I'm talking about, which comes under the Class D type of material devices. And just to let you know that the now in India also, it is separated from the the drug and pharma. It is now a separate regulatory guidelines called as Medical Device Regulation 2017, that is now followed, and it is a separate category. It does not come under the pharma.
Any other question from the participants? Okay. Thank you once again, sir, for knowledge sharing and sparing your valuable time. I request,
Thank
sir, to accept our token of appreciation in the form of a digital certificate.
thank you, doctor. I just
Thank you.
want to correct a spelling, so that probably you will do. Yeah,
Okay, I'll take care of it, sir.
yeah.
Thank you. Now I hand over the session to Professor Smita Kakad, madam, for further processing.
Thank you. Thanks, Dr. Santosh Chajet, sir. And thanks a lot, Dr. Akshay Shrivastava, sir, for very informative session with the actual guidance, with actual some examples on implantable medical devices for spinal cord regeneration. And this information is really helpful to us for designing some topics related to medical devices and implementable devices. In the morning, we have wonderful session by Dr. Sohan Chitlange sir to simplify the concepts in ranking framework about NIRF, ARIIA and AICT CII. Again, very helpful information provided by Dr. Rani Kankate ma'am in her session on establishment of IIC and role of MIC, Government of India. So it was a great day today, and new sessions on some interesting topic are waiting for us in coming 4 days. So with the permission of coordinator and respected principal sir, here I declare that today's session is concluded, and we will meet on the same platform tomorrow at 10 a.m. So bye everybody and have a good day.
Thank you. Thank you.